Hello. We are going to, in this video lesson, I want you to learn the different phases of mitosis and the major events that are happening. Now, one thing that we need to remind ourselves is what is the goal of mitosis? The goal of mitosis is to make two daughter cells each with the exact copy of chromosomes. So if you start with 46 chromosomes, each daughter cell needs to have 46 chromosomes each, and the same kind of chromosomes. You can't have extra copies of chromosome 2 and not have chromosome 1. So not only do we need the same number, but we need to make sure we have the same kind. And so you're going to watch for when and how the cell makes that happen. How does the cell organize the chromosomes? Because that's kind of the big event in mitosis. So let's first start by going over the different phases that you are going to expect to see. You're going to see interphase, and you can move the tiles here, and you're going to be asked to show that you understand these different tiles and different stages. So interphase would be the part of the cell that's not dividing, the part of the cell cycle that's not dividing. This cell is potentially in G1 or S or G2. The cell is not dividing in interphase. Things to make note of, we have a very obvious nucleus and very obvious cytoplasm. Now, the parts of the mitotic divisions would be prophase, metaphase, anaphase, and then telophase. And then we have our daughter cells, which are now in G1 as they're carrying out normal functions. Now, the ones up here, the reason this diagram shows two different cells, the ones up here would be plant cells. The ones on the bottom would be animal cells, plant cells on top, animal cells. And we'll talk about why there's a difference. So let's take a look. Now, you'll notice on your slides, almost every slide I have this GIF. It's a really handy GIF. It shows everything that's happening in a nice, almost like little animated section. And so you'll be able to reference this as we go on. So let's take a look at our first stage, and it's going to be on interphase. Now, again, technically, this is not mitosis. The cell is not dividing. And so, how do we know that? What are the clues in the pictures that allow us to say this cell is an interphase? So, let's take a look. We see a distinct nucleus, and we oftentimes see a distinct nucleolus. Now, this is kind of going back to first quarter stuff. The nucleolus, if you remember, is the dark area inside the nucleus. Um, so notice in our microscope slides, we see a nice, clearly defined nucleus, and we see also a darker nucleolus. And you see that with all of these microscope slides, this one's kind of hard to tell, but we see a nice, clearly defined nucleus. We also can't tell the chromosomes apart. All we see is this kind of mushy, mushy, indistinct chromosomes. And under the microscope, it just kind of looks like this vaguely gray stuff. And that's because the chromosomes are all nice and spread out. And so that is interphase. Now, as the cell starts to divide, let's take a look at prophase. Prophase is the first official stage in mitosis, and there's a lot going on. So let's break this down, and then we'll look at our microscope slides. The cell is going to break down the nuclear envelope. The nuclear envelope kind of breaks apart. Under the microscope, this just kind of is seen as the nucleus kind of gets bigger, less defined. We really can't tell where the nucleus starts and ends. The chromosomes, one of the biggest clues that a cell is in prophase, the chromosomes condense. They get really dark as the DNA winds up and becomes these really thick, condensed bodies. And what we see under the microscope is really dark chromosomes, and then the white areas as the chromosomes pull apart from each other. And so that's a big hallmark clue of prophase. Now, under the microscope, you don't see centrioles, but you can start to see the centrioles in under like electron microscopes. You can see the centrioles starting to move apart. We actually get two sets of centrioles, and they start migrating to the poles, opposite ends of the cells, like North Pole, South Pole. And so that's how you hear it. The centrioles migrate to the poles. They're going to opposite sides. Now, the centrioles are going to be building these spindle fibers, which you learned about in a previous lesson. Those spindle fibers are those long microtubules that attach to the chromosomes, and they're going to move the chromosomes about. They're going to uh, get attached and get ready to move the chromosomes. Now, why are we attaching to the chromosomes? Well, we want to organize them. So remember, I told you to watch out for that. So the next phase we're going to is metaphase. And in metaphase, the nuclear envelope is gone. And notice there's no organized nucleus. Like you cannot tell. In this cell that's highlighted here in this picture, and in this cell in the top right, and in the bottom cell right here, that is a cell, and you can't really tell where the nucleus is. That nuclear envelope's gone. Now, the chromosomes have been moved to the center line of the cell, and we call that the equator. And they're all lined up, and this is the cell organizing them. You can draw a line through that. Now, don't worry that some kind of look like they're going across where others are going up and down. That just means the cell is in a different alignment under the microscope. But the point is, you can see a line of chromosomes almost like a little set of soldiers going down. And if you notice that our cartoon picture, all of these chromosomes are lined up, and it's almost as if the cell is ready to say, hey, you chromosome number one, split, and one goes here, one goes there. Chromosome two, one goes there, one goes here. Chromosome number three, one goes to this daughter cell, one goes to this daughter cell. And they line up, and as they line up, what they're going to do in the next phase, they're going to split apart. Now, these are flexible chromosomes. We draw them like a letter X, but in reality, they can kind of move around a little bit. And so they're tethered at the center, that centromere that you saw in the other diagram. And as the spindle fibers pull them apart, they kind of collapse and kind of become almost like a little V shape as they get pulled apart. Now that's going to be the next stage, so I don't want to get ahead of myself, but notice what's the takeaway? They're all lined up. Now, this is often the shortest phase because once they're lined up, they're ready to be split. So notice our GIF right here, there's metaphase, and then they start pulling apart. And we call this anaphase. Now, an anaphase, under the microscope, it almost looks like taffy. Think about that Laffy Taffy candy. When you pull it apart, it kind of looks stringy. Well, the chromosomes get pulled apart. They're separated. And you can see the chromosomes now as they're getting dragged to the two sides, the two poles. Now, in anaphase, the nuclear envelope is gone still. The chromosomes are separated. We now call the chromatids, which were the individual copies, we now call them chromosomes. And we'll talk about that, and you will practice that. So, the centrioles are on opposite ends. and the chromosomes are going to be pulled to the opposite ends at this point. And that's what you see in this little GIF. The chromosomes separate and they're getting pulled, and now we're going to make two daughter cells there. Now it's the spindle fibers that are moving the chromosomes apart, and this is where the cell is organizing it, right? Each chromosome gets separated and pulled to the opposite daughter cell, and each daughter cell will have the same number and type of chromosomes. And again, we're going to practice that with a Jamboard. Um, and I'll walk you through it in the next video. Uh, so, what do we have at the end? We have telophase. Now, telophase, we have pulled our chromosomes apart. And so notice in our microscope slides, really dark, condensed, crowded packs. It's almost as if the cell is saying, I'm not going to lose you guys. I'm going to pull you together in two little groups. And that's very recognizable as our telophase. And so that is what's super, super helpful to notice. In each of these cells, you see this one set of chromosome and another set. And then we have an animal cell here, one set, and then the other set. And then we're going to have our two daughter cells. Now, the spindle fibers start breaking down, the nuclear envelope gets rebuilt, and the chromosomes kind of unfurl and uncoil once everything's done. And you start to see the cell looking like a normal cell, just maybe a little smaller. Now we get into cytokinesis, which is the cytoplasm dividing. And in cytokinesis, if you have a cell membrane in animal cells, the cell membrane pinches in, and we call it the cleavage furrow, where the cleavage means to split. So the cell membrane just pinches in, and we end up having two cells. So that's an animal cell. In a plant cell, we have a cell wall. And so we want to make a new cell wall, and we call that the cell plate. And you can start to see it kind of in this cell. It's starting to be made, and you'll see it in some of the microscope pictures that we find. Now, here would be all of these kind of in sequential. And so you can kind of see almost like a little flip book. You can see the cells um, lining up their chromosomes. Here's metaphase, and then splitting them in anaphase, and then grouping them in telophase. And then we have two daughter cells. And notice you can see the nucleus is really well defined. So here's our new cell wall in the onion root tip. Now, onion root tips are really classic locations to find cells dividing, because roots are growing through the soil. And so as the roots are growing through the soil, they're replacing and growing new cells. And these are just two different types of stains under the microscope, but you can see the chromosomes condensing, lining up in the middle, separating, and then there's our two daughter cells. Now you're going to have a chance to practice these. And so as you guys discuss with each other and talk amongst yourselves in class, you're going to then have a chance to practice these with the this slide, and also the MItosis lab that you're going to do. All right. So I hope that helps clear up what happens during mitosis.